Okay guys, so this is your review for chapter 3. I'm just going to go over some of the basics of the chapter and get into pretty much the overview of what we went over. So first off, I know we began talking about carbon. So carbon has 4 valence electrons, which means it's able to form 4 covalent bonds with other elements. So this is why carbon's so good at making these very big, very branched organic molecules. So it's very versatile just because we're able to form rings with carbon, we're able to branch carbon, or just form very, very long straight lines. So we talked about carbon forming single bonds, double bonds, triple bonds. If it has double bonds, it's probably going to be a more linear shape. And if it has just single bonds, well, we're going to have more of a tetrahedral shape. There's going to be some angles in it rather than it just being linear. So we went over that big chart, with our macromolecules, with the proteins, the lipids, the nucleic acids, and the carbohydrates. We did the structure, we did the function, the monomers, the polymers. Make sure you guys know that thing inside and out. Know which elements are in each of our structures, okay? Look at your R groups for the proteins. See what elements you see in there, okay? This will help you out on the test just so you can differentiate between the structures of each of those macromolecules. And then again, make sure you know your monomers and your polymers, okay? We went over the dehydration and the hydrolysis reactions. Let me do one of those with you guys real quick. If we have a dehydration reaction, so I don't know, let's say we have monomer plus another monomer, we're going to get a polymer out of it, and we're also going to get water. Why are we getting water? Well, because on this monomer and this monomer, we are going to have side groups that are OH or HO, however you want to say it, hydroxyl group. And then we have a hydrogen on the other side. These two right here are going to combine to form our water. And they break off, guys, in order for these monomers to bind together right here and get our polymer out of it. So again, guys, this is a dehydration reaction, dehydration synthesis reaction, condensation reaction. It's all the same. The other one was the hydrolysis reaction. This is where we have a polymer, and we are going to break it into maybe still a polymer, but we break a monomer off, okay? In order to do that, we need to add H2O wherever that bond is. So in our reactants on this one, we are going to have H2O, all right? Now, let me show you something here, guys, real quick with hydrolysis reactions. Let's say we have 2, 3, 4, 5. We'll go with 7. Okay? If I ask you a question with how many water molecules am I going to need to take, let's say, 4 monomers off of here, break 4 monomers off, and we want them to be individual monomers, okay? If I say I'm going to break them into 4 monomers, obviously I don't just want to break 4 of them off because they're not monomers. If I break, like, let's say I'm going to take these 4 here. Here, I'll color them in a little bit for you guys. If I take 1, 2, 3, if I take these 4 off and I just break it right here, okay, I got a polymer on this side and I got a polymer on this side. I don't have any monomers there, okay? So I'm going to break these 4 off and we're going to break them into monomers. So I still want my 3 here, okay? But here's what we have to do. We have to count our bonds. So I have 1 bond here. That's going to break these 2 apart. 2 bonds. Okay, so if I do this, I'm just kind of draw arrows here, guys. There's our monomer. He's broken off. If I break bond 2, there's our monomer. If I break bond 3, there's our monomer. Okay, so I have my 3 monomers here. Okay. So I broke 3 bonds. I have to break 1 more. I have to break this bond here, number 4, in order to get my last monomer off. Okay? So, in order to get, if I go back to the products up here, that I need to break 4 bonds. So how many water molecules am I going to need? Well, I'm going to need 4. Okay? Watch how that is different than this, though. I'm going to use my same amount in my 1, 2, 3, 4, 5, 6, and 7. Same amount in my polymer here. Now, if the question says, hey, I want to break this into, break apart this chain completely into individual monomers. Okay, we're breaking apart completely. So let's count our bonds up. So I have 1, there's 1, 2, 3, 4, 5, and 6. Okay, so if I'm going to break it completely apart, I have to break 6 bonds so that I get my 7 polymers, all completely, disjoined. So since it's like this, guys, I'm only going to need 6 H2Os because I only broke 6 bonds. I need to break 4 up here, okay, in order to get those 4 off. But down here, I'm breaking all of them. All right, so if you're breaking the whole thing apart, guys, it's 1 less than the number of monomers you have. If you're not breaking the whole thing apart, whatever that number is, I'm breaking off 4, I'm going to need 4 waters, okay? So there's your difference between if we break it all apart or just break a little bit apart. Let me just erase it this way. There we go. Okay. Okay, so we went over that. We're good there. Make sure you know your difference between like cellulose and chitin, okay? Those polymers. Next thing, guys, we went over saturated and unsaturated fats. Saturated fats, if you remember, they are solids at room temperature, usually come from animals. And unsaturated fats, they're going to come from plants. They're going to be your liquids. Okay. Difference in structure, unsaturated are going to have at least 1 double bond. Saturated fatty acids, no double bonds. Unsaturated fatty acids, because of that double bond, we're going to have a kink in there somewhere, so it's going to kink just a little bit. And the saturated fatty acids is going to be more linear. There's also hydrogenated oil, if you guys have ever heard of that. What that is it's at one time it was a unsaturated fatty acid, okay? So what they did was they took this unsaturated fatty acid and hydrogenated it. In other words, added hydrogens to it. What does that do? Well, that's going to get rid of our double bond there, and that's going to keep it as solid at room temperature. So kind of cool. That's how they do some of like the butters with like vegetable oil and stuff like that. All right. Make sure you guys know your structure of a protein. Make sure you know what functional groups are on both sides. Okay. Make sure you know the R group is up top. Everything is connected to a central alpha carbon. Let me go back to our chart here. So we are also going to have our nucleic acids. We went through the apple in the tree and the car in the garage. Okay, so there's also 2 terms for our different nucleotides. We have pyrimidines and we have purines. How I remember this, guys, is pure agony. All right, pure agony. All right, purines, pure. Agony begins with AG. So your purines are adenine and guanine. Okay, so that's how I remember that one. And then all of your other nucleotides are going to be pyrimidines. This is going to be our cytosine, our thymine, and then for RNA, our uracil. Okay? One more thing to go over with DNA, guys. If we have a strain of DNA, one side of it, we're always going to have a 5' and a 3'. So if we did like A, T, T, G, C, T, A, G, G, T, A. Okay? The other side would read, so A goes to T, apple in the tree, T goes to A, okay? So your primes, guys, make sure we have opposite primes, all right? And we could write this second one either way, guys. If we wanted to write it the opposite way, you know, on a test, there's not like any right way to write it here. Okay. If it's just alone, we can write it either way, guys. But when it's bonded to our other helix, we're going to write it that way, as I have up here, just because in the middle, the red lines there, those are our hydrogen bonds to make our double helix, to make our opposite anti-parallel sides for our DNA. All right. Okay. So I'm going to get some Petri dishes here, and we'll bring them. There's our Petri dishes. Okay. All right. So we're going to say a C for carbohydrates, a L for lipids, a B for protein, and then NC for nucleic acid. Okay. Now let's say that on these Petri dishes we put a medium and it has carbon-14 on it. Okay, a radioactive isotope of carbon. And what these would look like, guys, is it'd be like a gelatinous material in these Petri dishes, and maybe a little bit of like a broth on top. Okay, but anyway, so we are going to put in this one some carbohydrates on top of our C14 broth. On this one, we're going to put some lipids. On this one, we're going to put some proteins. And the last one, we're going to put nucleic acids. Okay, what we're testing for is to see if we have carbon in this one, this one, this one, and this one. Well, if you look at your structures of each one of these, macromolecules, you will see that carbon is in each one of them, okay? So over time, what would happen, guys, is as whatever cells we're putting on there with the carbohydrates, with the lipids, and so on and so forth, what we would see is that over time, they would use this carbon-14 on here, and they would bring it up, all right? So they would use it. So what we would have, all right, we would have some growth, okay, on our plates here, because every single one of these contains carbon. All right. Let's say we did the same thing, and I'm going to draw Petri dishes down here. This one's still going to be carbohydrates. This one's still going to be lipids, still proteins, and still nucleic acids. Okay, cool. So let's use a different medium. All right, let's use N15. An isotope of nitrogen. Okay. All right, did the same technique, guys. And we are going to see which ones are going to have growth. Which ones are going to take up that N15 isotope. And they're only going to take up N15 if they have nitrogen in that monomer or that polymer, okay? So what we would find here is that you guys know carbohydrates, they do not have nitrogen in them. They're just, CN, H2N, ON. Okay, those N's are just our numbers. Those N's are not nitrogen. So it'd be like C6H12O6. For this one here, okay, you know it's like the same thing almost. It's like, you know, C10H20O2, something like that, okay? Our proteins and our nucleic acids, the proteins have that amino group on the side that has the N, and the nucleic acids have the nitrogenous base that has the N. So these ones, we would have growth. Okay? And then the carbohydrates and the lipids would have no growth because they don't have any nitrogen in them. All right? So there is a good example there, guys. And then the last thing I wanted to go over with you guys is our monomers of carbohydrates, which we have 3 of them. We have glucose, fructose, and galactose. So for all 3 of these guys, our chemical formula is C6H12O6. Okay, so how are they all the same, but they all have different names? Well, the way they're put together is different. The way our elements are arranged in these 3 is different. So even though they have the same chemical formula, the structure is different. And we call these structural isomers because their structure is different even though they have the same amount of elements in the same ratio. All right, guys, so that is it for the review for Chapter 3. I think we went over, you know, everything in pretty good detail there, all the important stuff. Make sure you study your chart, you know, your monomers, your polymers, your structure, your function of all of the macromolecules. And then study for your Chapter 3 quiz, guys. Have a good rest of your day.